genes are very often controlled by extracellular signals. In the case of bacteria, these signals typically mean molecules present in the growth medium. So growth medium में जो molecules हैं वो ultimately इन genes को signal करेंगे और फिर इन signals के लिए इन gene को control करेंगे. These signals are communicated to the genes by regulatory proteins which come in two types: positive regulators or activators and negative regulators or repressors. Typically, these regulators are DNA binding proteins that recognize specific sites at or near the genes they control. An activator increases transcription of the regulated gene and repressor decreases or eliminates that transcription. How do these regulators work? First, RNA polymerase binds to the promoter in a closed complex in which the DNA strands remained together. That is, they are in the form of duplex. The polymerase promoter complex then undergoes a transition to an open complex in which the DNA at the start site of transcription is unwound and the polymerase is positioned to initiate transcription. This is followed by promoter escape, the step in which polymerase leaves the promoter and starts transcribing. Polymerase then proceeds through the elongation phase before finally terminating. Which steps are stimulated by activators and inhibited by repressors depends on the promoter and regulators in question. The most common step at which gene expression is regulated is the initiation of transcription. There are two reasons why this might make sense. First, transcription initiation is the most energetically efficient step to regulate. By this we mean that deciding whether or not to express a gene at the first step ensures that no energy or resources are wasted. Second, regulation at this first step is easier to do. There is only a single copy of each gene, and so typically only a single promoter on a single DNA molecule must be regulated to control expression of a given gene. Why then is not all regulation focused on this step of transcription initiation? Regulating later steps can have two advantages. First, it allows for more inputs. If a gene is regulated at more than one step, more signals can modulate its expression, or the same signals can do so even more effectively. Second, regulation at steps later than transcription initiation can reduce the response time. Thus, consider again the example of translational regulation. If a signal relieves repression of this step, the protein product encoded by the gene will be produced immediately upon receipt of this signal. This reduced response time might obviously be advantageous in some situations. Many promoters are regulated by activators that help RNA polymerase bind DNA and by repressors that block that binding. At many promoters, in the absence of regulatory proteins, RNA polymerase binds only weakly. This is because one or more of the promoter elements is absent or imperfect. When polymerase does occasionally bind, however, it spontaneously undergoes a transition to the open complex and initiates transcription. This gives a low level of constitutive expression, which is also called basal level. Binding of RNA polymerase is the rate limiting step in this case. To control expression from such a promoter, a repressor need only bind to a site overlapping the region bound by polymerase. In that way, the repressor blocks polymerase binding to the promoter, thereby preventing transcription. Although it is important to note that repression can work in other ways as well. The site on DNA where a repressor binds is called an operator. To activate transcription from this promoter, an activator can just help the polymerase bind the promoter. Typically, this is achieved as follows. The activator uses one surface to bind to a site on the DNA near the promoter. With another surface, the activator simultaneously interacts with RNA polymerase, bringing the enzyme to the promoter. So here you can see this is activator binding site, this is operator region, and this is enzyme. So if no activator is attached, the transcription will take place at the basal level, that is the constitutive expression. So if a repressor binds to this operator region, no transcription takes place. And in the third case, an activator is bound at this activator binding site. It interacts with the RNA polymerase on the other side and transcription will take place. This mechanism, often called recruitment, is an example of cooperative binding of proteins to DNA. The interactions between the activator and polymerase and between activator and DNA serve merely adhesive roles. The enzyme is active and the activator simply brings it to the nearby promoter. Once there, it spontaneously isomerizes to the open complex and initiates transcription.